Hello, I'm Jane Morris, and today I'm going to cover the risk assessment of genetically modified herbicide-tolerant maize for food and feed use, with special emphasis on its safety for people in southern African countries. We'll briefly cover, firstly, what types of herbicide-tolerant or HT maize are on the market, how do they work, where is herbicide-tolerant maize grown, and how do we assess the risks for food and feed. We'll then go through the full risk assessment for one particular type of herbicide-tolerant maize as an example. Up to now, herbicide-tolerant genetically modified maize is only available against 4 herbicides. The first and most common is glyphosate, otherwise known as Roundup. Then there's glufosinate ammonium, most commonly marketed as Basta. Both of these are broad-spectrum herbicides. Then there's 2,4-D, which is selective for broadleaf weeds, and more recently dicamba, which is similar to 2,4-D. Glyphosate targets an enzyme in the pathway for synthesis of aromatic amino acids. The enzyme has a very long name, but it's normally just shortened to EPSP synthase or EPSPS. The first glyphosate-tolerant plants were made using an EPSPS enzyme that was modified to prevent binding of glyphosate. But second-generation plants used an enzyme from the soil bacterium Agrobacterium. This bacterium, with an EPSP synthase resistant to glyphosate, was isolated from a waste stream at a glyphosate production facility, and the enzyme was cloned into maize. Glyphosate tolerance is the most prevalent form of herbicide tolerance. Tolerance to glufosinate ammonium, or glufosinate for short, is probably the second most common form of herbicide tolerance. Glufosinate is also known as phosphinothricin. It has a similar structure to glutamic acid, and it inhibits the enzyme glutamine synthetase, which is essential for nitrogen metabolism. 2 genes from Streptomyces, called PAT and BAR, have been cloned into plants to produce an enzyme that acetylates and inactivates the herbicide. For 2,4-D tolerance, there is only one approved event in maize. It contains a gene from the soil bacterium Sphingobium herbicidovorans, expressing the enzyme RL-oxyalkanoate dioxygenase, known as AAD1, that catalyzes the breakdown of the herbicide. Then we come on to the 4th herbicide, dicamba. It works similarly to 2,4-D by acting as a plant auxin to stimulate abnormal growth. Dicamba-tolerant maize, developed by Monsanto, has only been approved in the USA in 2016 in combination with glufosinate. It contains a microbial gene coding for an enzyme that uses dicamba as substrate. Since South Africa is already quite well advanced in its use of GMOs, it's useful to know what HT maize events are already approved. South Africa has a system called commodity clearance, whereby GMOs can be approved for use as food or feed, but not yet for cultivation. A number of these events approved for commodity clearance are for imported maize, which may be further exported to other countries in the region, often being milled first depending on the requirements of the country concerned. HT events have been approved against glyphosate and glufosinate as well as 2,4-D. Many of these approved HT maize events are also available in stacks with other genes, most often pesticidal Bt genes. You can view the separate presentations on Bt maize for more information. 3 of these HT events are fully approved for commercial cultivation in South Africa. In order to work through a risk assessment of Bt maize for food and feed, we need to remember once again the normal procedure for risk assessment. The steps are, firstly, hazard identification, that is, what might go wrong. And note that this is not the same as what might change. We then look at how serious would the consequences be, that is, what would be the harm. How likely is it to occur? And finally, we estimate risk as a combination of harm and likelihood. A decision is then made on the basis of acceptability of the risk, and at this stage we may also take into account the expected benefits. So, for herbicide-tolerant maize, we can ask the following questions to help us identify any hazards that relate to food and feed safety. Firstly, is the DNA stably inserted with a single site of integration? That's important in terms of any unexpected effects. Is the protein it codes for toxic to humans or animals? Is the protein allergenic? Are there any changes in composition of the maize? And are there any changes in anti-nutrients such as phytate or protease inhibitors? Could there be any long-term effect from eating the herbicide-tolerant maize? And finally, because this is an environmental issue rather than a food safety issue, would it be a problem if unmilled maize grain fell from trucks and or was planted by farmers? We're going to follow these questions through the risk assessment process with one interesting example, maize NK603. I chose NK603 as an interesting example because there's been a lot of controversy, as it was the subject of a discredited long-term feeding trial conducted by Seralini and his colleagues in France. Linked to the Seralini study, there is also an ongoing debate about the safety of the herbicide glyphosate, or Roundup, which was deemed to be a probable carcinogenic hazard by one UN agency, but subsequently determined to be unlikely to be a carcinogenic risk by another agency. And this highlights the need to examine the safety of GM crops in combination with chemicals that are applied to the crop. We start by looking at the inserted genes and their stability and expression. NK603 contains two glyphosate-resistant EPSPS genes from Agrobacterium strain CP4, inserted by particle bombardment. Each has a different promoter sequence, one the rice actin promoter, the other the more commonly used cauliflower mosaic virus 35S promoter. Both are fused to chloroplast transit peptide sequences, since EPSPS is located in the plant chloroplast. To give you more background on EPSPS, it is found in plants, bacteria and fungi, and it's involved in the synthesis of the amino acids tyrosine, phenylalanine and tryptophan. It is not found in animals which do not synthesize their own amino acids. A single copy of each gene cassette was inserted, and bioinformatic analysis showed no other unintentional open reading frames. There were some other DNA fragments integrated, but these did not result in any changes in gene expression that could be detected. The inserted genes were stable over many generations, and the mean level of expression in maize grain was 10.9 micrograms per gram fresh weight. Next, we look at toxicity and allergenicity. CP4 EPSPS is from a family of proteins with a history of safe consumption, so no toxicity or allergenicity was expected. Acute toxicity studies are normally carried out with the isolated protein because it's not possible to get a sufficiently high level of intake with the whole feed. So an acute oral toxicity study with the isolated EPSPS in mice showed no effect at 572 milligrams per kilogram body weight. Bioinformatics sequence analysis showed no homology with known toxins. CP4 EPSPS is also rapidly degraded in gastric fluid, whereas allergens are normally stable. Bioinformatics sequence analysis showed no homology with known allergens at the 8 amino acid sequence level. We then go on to see if there are any compositional changes. This is done by comparison with the near isogenic conventional maize line, bearing in mind the normal compositional range of maize varieties in general. Useful information on maize composition is available in the OECD consensus document, and I give the link to the document here. Maize does contain some anti-nutrients, particularly phytates, raffinose and trypsin and chymotrypsin inhibitors, so it's important to check for any changes in the levels of these compounds. NK603 and its near-isogenic conventional counterpart were grown in 5 locations in the United States, and the grain composition was compared. 4 trials were then carried out in the European Union that included comparison with 19 conventional hybrids, that is 5 hybrids per site, with 1 hybrid being planted at 2 locations. The NK603 plants were treated with Roundup. Proximates, fibre, minerals, amino acids, fatty acids, vitamin E, anti-nutrients, that is phytate and trypsin inhibitor, and secondary metabolites were all measured. There were some variations in composition, but all values were within published literature ranges for maize and within a 99% tolerance level for the conventional hybrids in the trials. So, in conclusion, we can say that from these studies, no hazards were identified that would make the maize unsafe to eat. A recent paper, published by a team, including the French scientist Seralini, whom I've already mentioned, has however implied that the compositional analysis carried out previously was inadequate. They undertook metabolomic and proteomic analysis, and they've suggested that there are a number of metabolic perturbations in NK603 and that there are increases in polyamine levels such as the nasty sounding compounds putrescine and cadaverine, which, by the way, are found in most foods and have a number of beneficial effects as antioxidants and are necessary to support normal metabolism. Polyamines are, however, not mentioned in the OECD consensus document on maize, and there's a lack of information on natural variation in their content. It is known that their levels can vary considerably and that they are produced in plants in response to stress. This recent paper has been criticised on a number of levels. Firstly, there was a lack of replicates, and the plants were not grown under controlled conditions, so there would be variation simply from the growing conditions. One result of the lack of controlled growing conditions was that there were differences in fungal contamination between NK603 and the control, which would confound the analysis. Then there was only NK603 and its supposedly near-isogenic conventional counterpart grown, so there were no other conventional lines grown that would show the complete range of natural variation. Then the so-called near-isogenic line was probably not actually properly produced by backcrossing to a recurrent parent, so the comparison may not be realistic. And finally, the paper completely ignores 2 other recent publications that give results of analyses carried out on NK603 and its near-isoline, and that contravene the conclusions in this one. The paper by Venkatesh et al. Found that growing location and the tester used in hybrid formation had a markedly greater effect on composition than did the GM trait. And then there's a paper by Harrigan et al. Using metabolomics analysis, where they showed that variations were more likely due to growing conditions and or associated with minor genomic differences due to conventional backcrossing practices than were likely due to variations from the GM trait itself. But as a general conclusion, we can say that there is no reason to associate any of the observed variations, whether or not they are due to the genetic modification, to specific safety concerns. But what about longer-term feeding studies as an indication of safety? As I described in the presentation on food safety of BT maize, the European Food Safety Authority recommends longer-term feeding studies only when specific hazards are identified to justify them. But despite this, the EU regulations require 90-day rat feeding studies for all applications. In fact, a number of feeding trials were carried out for NK603. These include a 42-day poultry feeding study, and a 103-day pig feeding study. Although not shown here for lack of space, there have also been some cattle feeding studies and another pig feeding study. All the feeding studies showed that NK603 gave similar results to conventional maize. The results of the 90-day rat feedings trial also indicated comparable results between NK603 and the conventional counterpart, and this was verified by the European Food Safety Authority. I have also looked at the raw data, and although I'm not a statistician, I was satisfied with the outcome. The team of the French scientist Ferrellini were involved in the omics study on NK603 that I discussed earlier, and prior to that study, Ferrellini's team reanalyzed the data from the 90-day feeding trial and suggested that there were effects in the kidney and liver. To follow up on this, they undertook a 2-year rat feeding trial comparing rats fed NK603, NK603 with glyphosate, glyphosate alone, and rats fed conventional maize. The methodology was sharply criticised and the paper retracted. It was subsequently republished elsewhere without further peer review. Ferrellini concluded that NK603 and glyphosate at low levels both induced severe adverse health effect, including tumours. The study was criticised on the basis of poor study design, poor choice of rat strain. He used Sprague-Dawley rats that are prone to tumours in any case. There were also not enough animals to draw proper conclusions, there was inadequate statistical analysis, and his conclusions were not supported by the data. While the media and anti-GM activists have been quick to jump on the results of this study, and now also onto the results of his latest study indicating metabolic perturbations, I should point out that European and other authorities have continued to permit NK603 to be used for food and feed. NK603 has been in the human food chain for about 15 years with no adverse effects reported. Besides NK603 itself, Ferrellini also suggested that glyphosate was carcinogenic. This was followed up by a report in 2015 from the WHO International Agency for Research on Cancer, the IARC, who published a report saying there was limited evidence of human carcinogenicity. They stated that it was probably carcinogenic, i.e., that there was a hazard. What has not been realised by many people is that foods like grapefruit juice and apples fall under the same classification. The IARC does not assess the actual risk that relates to the dose levels. Subsequently, a FAO and WHO report has concluded that it is unlikely to be carcinogenic to humans at anticipated dietary exposures, and that it is not carcinogenic in rats, i.e., there is a low risk. This report gave acceptable daily intake levels as up to 1 mg per kilogram body weight, although actual genotoxicity was not seen even at levels of up to 2,000 mg per kilogram. The allowable maximum residue levels for glyphosate in maize are 5 mg per kilogram under the International Codex Alimentarius, although the limits may differ in some countries. In practice, the residues are most often much lower than this. Even if all the maize consumed contained the Codex's highest maximum residue level, a 60 kilogram person would need to eat 12 kilograms of maize per day to get to this level of intake, which would be well nigh impossible. Of course, we need to take into account that in southern Africa, maize is a staple food, unlike in Europe or the States. Of all the countries in the region, Lesotho has the highest consumption at 174 kilograms per person per year. However, the acute toxicities tests showed no adverse effects at consumption levels more than 1,000 times greater than the daily consumption. The 90-day feeding trials showed no adverse effects that could be ascribed to NK603 at the maximum dosage, and this was confirmed in a range of other feeding trials. The 2-year feeding study purporting to show adverse effects has been widely discredited, and this means that the likelihood of adverse effects, even in the long term, is negligible. So we can reach the overall conclusion that the risk for food and feed use is negligible and NK603 is safe for people in southern Africa to eat. So now, at the end, we consider any environmental risks if some grain is spilt, or if farmers retain and plant some of the grain. Of course, this is only an issue in the case of whole grain, not milled grain. We should bear in mind that NK603 is already cultivated in South Africa, so it is present in the region. It's unlikely to have any competitive advantage over conventional maize, except where glyphosate is sprayed. Maize also doesn't have weedy or invasive characteristics, and it has no wild relatives in southern Africa. In any case, any volunteer plants could be easily killed with other herbicides. If farmers planted the maize, it would be as safe to eat as conventional maize. It is in any case an annual crop that can be easily controlled, and it would lose hybrid vigour over successive seasons. So, as a final conclusion, we can say that NK603 can be safely used in the SADC region for food and feed. This is just one example of a risk assessment, and each event needs to be assessed on a case-by-case basis. As a follow-up to this presentation, I give an example of a different herbicide tolerance maize in my presentation on risk assessment of herbicide-tolerant maize for environmental release, and you might wish to have a look at that presentation. Thank you.